This screencast looks at mutations, different types of mutations that happen in DNA. Those mutations that happen in DNA are called point mutations. Okay, and mutations are mistakes that occur when DNA is copied. So DNA replicates, as we know, as it goes from cell to cell, and when a mistake is made, that is called a mutation. Okay, they occur naturally all the time. But there are certain things that increase the rate at which mutations occur, radiation being one of those, and also chemicals that which we call mutagens could also increase the rate at mutations occur. And this links in with the smoking bit that we looked at back at f215. We looked at lung cancer and how that lung cancer is often a result of chemicals in tar, in cigarette smoke, which we call carcinogens, which cause mutations in the DNA of lung tissue and result in cells growing out of control and forming a tumour. Okay. Anyway, looking at the different types of mutation that we have. Okay, the first type is point mutations, which is when we substitute a base for another. These are sometimes called substitutions. Okay, so one base gets swapped for another one. Okay, which is a mistake that occurs when DNA is copied. Okay, the other type is an insertion mutation, which is when a base is added. Okay. And then there's another type which is called a deletion mutation, which is when a base is removed. Okay. Now, insertion and deletions together are sometimes called frame shift mutations because they completely disrupt the triplet code on DNA. Okay, and the subsequent messenger RNA, which completely changes the sequence of amino acids, which has a very dramatic effect on the protein structure and therefore probably its function. Okay, point mutations, which are substitutions, only alter at worst one amino acid, but sometimes it doesn't even alter the amino acid at all. If you remember when we looked at the idea of the degenerate code, is because the triplet codes for that are shared for the same amino acid are similar. It is very possible for a substitution mutation to change a base for another base, and that other base changes the code into the code for the same amino acid. Okay, so there wouldn't be any change at all. Okay, so sometimes there is no change even with a point mutation, but at worst it's only going to change one amino acid. Whereas frame shift mutations alter the whole triplet sequence, and this would alter the whole polypeptide sequence and usually have a dramatic effect on structure and therefore function of the protein. Illustrating some of that with a diagram below. Okay, so here I've got a normal sequence of bases along here. Okay, so here are the DNA bases and the RNA transcripts, and then what those translate to in terms of amino acids. Okay, so that's a normal setup here. Here I've got an example of a substitution, so if we look along here we can see that the C here has been changed to a T here, which creates that triplet code TTT instead of CTT. Okay, that transcribes to AAA rather than GAA. Okay, and that is the triplet code, or the codon, which translates to the amino acid lysine rather than this amino acid here. Okay, so that has changed just one amino acid. Okay, so substitutions at worst change one amino acid, and it would have been very possible that the substitution could have created another triplet code, okay, which could have transcribed through to a codon which actually coded for the same amino acid, so there might not have been any change at all, but in this case there has just been a change of one amino acid. It doesn't affect the integrity of the rest of the DNA. Okay, so this triplet code here is the same as that, and therefore the same amino acid here, and similarly this is the same as that, and therefore same amino acid there. Okay, in terms of these next two, insertions and deletions, which together we sometimes call frame shift mutations, is that what we've done here, we've inserted a C in here, so the CT, okay, moves along one, okay, and the result of this is that this transcribes into a different codon, and so does this, okay, which results in two new amino acids, and all amino acids which would carry on from this insertion would be different, okay, and therefore be a very different primary structure for that polypeptide. Similarly, a deletion, okay, which is when we've taken out this A, okay, well this A here from the normal, we've taken one of those out, so again we get a completely different triplet code here, which leads to three different amino acids occurring here, and again a very different polypeptide from the original one, and therefore presumably a very different function. Okay, so those are all the different types. Okay, and below I've got a little animation to show that as well.
A point mutation is a change in the nucleotide sequence of a gene. This can result from an error during replication that is not corrected by the usual repair mechanisms. Under normal circumstances, adenines and thymines should bind to one another, as should cytosines and guanines. In a base substitution mutation, a non-complementary nucleotide is incorporated into the new strand. The resulting altered triplet may code for a different amino acid, or even a stop codon. Here we see 4 codons and their corresponding amino acids. A change within the second triplet results in a change in the amino acid. None of the other amino acids are affected. Insertions and deletions result in frame shift mutations. The insertion of an extra nucleotide not only affects the triplet it is part of, but all subsequent triplets, thus drastically altering the protein product. The result is similar if a nucleotide is deleted.
So just summarising, going back to the top here, so we've got different types of mutation. We've got substitution mutations, okay, we've just substituted one base for another, which at worst changes the one amino acid in our polypeptide. We've got insertion mutations, which is when we add a base to our code, which changes all the triplet codes and therefore has a dramatic effect on our polypeptide. And deletion mutations, similarly, removing one base affects all the triplet codes. And insertion and deletion mutations are sometimes known as frame shift mutations. Okay, the other thing to remember is the business about degenerate code. Okay, so for a substitution mutation, it's possible to change one base for another base, and that's to result in exactly the same amino acid being produced in the protein product. Okay, so that's moving on to the next slide. This slide looks at the effects of mutations on protein function. Okay, so effects on protein can either be beneficial, providing benefit for the protein function and therefore the organism, okay, or it could be harmful, or it can be neutral. Okay, let's look at all of those in turn. Okay, so beneficial. The mutation could make the protein work more effectively for the individual. Okay, so for example, the mutations that occur in bacteria, okay, which produce enzymes. Sometimes you get mutations which can produce a protein which breaks down an antibiotic molecule, conferring resistance to that antibiotic on the bacteria, okay, and therefore that bacteria very quickly multiplies in the population and becomes the normal strain of bacteria, and you end up having a population of antibiotic resistant bacteria. Okay, an example in humans, okay, so humans evolved from first humans were found in equatorial Africa, okay, where there's lots of sunlight on the equator, okay, so that they tended to have dark skin, Okay, the reason that they had dark skin was to protect themselves from sunlight. Okay, but the other factor is that vitamin D, which is produced by the skin, their dark skin wouldn't necessarily be hugely efficient at creating vitamin D, but because there was lots and lots of sunlight, it didn't really matter, in that the darkness of their skin wasn't a limiting factor in producing vitamin D. Okay. However, as humans migrated to north and south, to less sunny regions, suddenly the production of vitamin D becomes a selection pressure. Okay, and therefore any mutation to provide paler skin would become beneficial. Okay, so in less sunny regions, they don't need the dark skin to protect themselves from sunburn. Okay, but they do need the paler skin to absorb sufficient sunlight from the limited sunlight around to create sufficient vitamin D, which is important for absorption of calcium from the gut. Okay, the exception to this, just as a complete aside, are Inuits. Okay, they didn't have the same selection pressure for producing vitamin D because they get their vitamin D from the fish in their diet. So Inuits that live in Greenland and near the North Pole, okay, still have quite dark skin, but they don't get their vitamin D from sunlight. They get their vitamin D from fish in their diet. Okay, harmful mutations. Okay, so these are mutations that result in the protein, okay, not working particularly well, and therefore conferring some form of harmful effect on the individual. Okay, so for example, the mutation that leads to cystic fibrosis would be an example that creates a harmful effect on the individual, okay, and confers less chance of survival on that individual. Okay, and then there are neutral mutations that result in a new allele but has no influence on the survival of that organism. Okay, so the point mutation might create no change in the amino acid sequence. This goes back to the idea of the degenerate code, which we mentioned before. The case of mutation could result in exactly the same amino acid and therefore the same primary sequence, secondary folding and tertiary folding, and therefore the same function of the protein. Okay, some changes in the polypeptide sequence have no influence on protein function. Okay, so you might have a change in the polypeptide change in the primary sequence, but because that change might be away from the active site, let's say, of an enzyme, and therefore it doesn't actually affect the way that enzyme works, even though it has a change perhaps on the overall protein shape, it doesn't actually affect the protein function. Okay, and then some changes in protein function have no effect on survival. Okay, so a change in, let's say, hair colour, for example, might have no effect on the survival of the organism. Okay, so although it has changed the phenotype of the organism, it hasn't necessarily changed its survival. Okay, so those could all be what we might call neutral mutations. Okay, so diagrammatically looking at that below. Okay, mutations creates variation, which is hugely important for evolution. Unfavourable mutations are selected against, so some sort of unfavourable mutation, okay, selected against, so they tend not to survive and don't produce progeny. Okay, reproduction and mutations continue to occur with harmful mutations not producing offspring, and unfavourable mutations more likely to produce offspring. Okay, and they keep reproducing. Okay, and that's hugely important in the whole idea of evolution. Okay.